So hello students, today I'll be explaining about implantation. So implantation, what all different steps are present in implantation or that will be explained. First of all, what is the definition of implantation? Implantation means what? So here implantation is attachment of blastocyst to the endometrium of the uterus, or you can tell burying of blastocyst to the endometrium of uterus. So it is human implantation. So rather here attachment of blastocyst to the wall of endometrium. To the wall of endometrium is called implantation. Is called as implantation. So another way of defining will be, instead of telling attachment here, you can tell burying of endometrium into the wall of, burying of the blastocyst in the wall of endometrium. So this becomes the definition of implantation. Okay, so implantation basically is, so where does it implant? You'll know that in female reproductive system, so this is fimbriae, this is the uterus and the vagina. So if you consider only the half portion here, so to the, this will be attached to the ligaments. So here, as the zygote divides and forms an early embryonic stage called blastocyst. So zygote is formed here in the ampullary region. So zygote is formed in the ampullary region, and as it divides, it is going to form one early embryonic stage, and that early embryonic stage is blastocyst. The blastocyst will bury in this wall. It means that the wall of the uterus have three layers: the perimetrium, the middle myometrium, and the inner endometrium. This blastocyst will go and attach the endometrium, or you can tell bury itself in the endometrium. So this one, the innermost layer here, it will be called as endometrium. So this blastocyst will bury in the endometrial wall or attach itself into the endometrial wall, and this phenomena is called as implantation. So this completes the definition of implantation. Now next is, what are the various events that lead to this implantation? For that, I'm going to draw a diagram here. So what happens is, this is a fundic region of the uterus. Again, as usual, this is what? The fimbriae, the ampulla, the broadest part, and the narrowest part here will be the isthmus. And then comes the uterus. And this will be the cervix, and this is going to be the vagina. So here, this portion, this is the wall of the endometrium, wall of the uterus. And this will be the fallopian tube. So this is nothing but the wall of the uterus. So here also this is going to be the wall of uterus. So this is going to be the wall of uterus. So here this wall is going to have three layers. The outer layer will be called perimetrium, where here it is not differentiated since we are not dealing with the walls of the uterus on today's topic. So the uterus will be divided into three layers. The outer layer is called perimetrium, the middle layer is called myometrium, and the inner layer is called as endometrium. That means this inner layer, this is called as the endometrium. So in this endometrium, the blastocyst is going to get attached or buried. Therefore, that phenomena is called implantation. Now, which all events that leads to implantation will be explained now. So you all know a process occurs, reproductive process called ovulation. Now, what is that ovulation is? The ovum here will be released outside the ovary. Okay, even though it may not be the proportionate to the actual size here. So this ovum is going to be released outside the ovary. This phenomenon is called as ovulation. Once the ovulation occurs, that is after ovulation occurs, the fimbriae, finger-like projections, they are going to collect this ovum. Okay, so they are going to take this ovum and direct it to inside the fallopian tube. Now, ovum is collected by fimbriae or swept up by fimbriae. So when ovum is so so now consider this is the ovum. Okay. Now ovum is going to reach to the ampullary region. This is the widest part actually. This part is called as ampullary region of fallopian tube. Here the ovum will be fertilized with a sperm. After the haploid sperm fuses with the haploid ovum, the resulting cell will be called as the diploid zygote. It will be called as the diploid zygote. So diploid zygote, it means zygote is unicellular. It's not a multicellular entity. It is a unicellular entity. Okay. So diploid zygote is formed. And by this, what you'll come to know, which I told last time itself, fertilization restores the diploidic condition in the zygote. It means zygote remains in a diploid conditions. So this is regarding formation of zygote. Now consider this is the zygote that is formed. What will occur to what happens to the zygote once it is formed in the ampullary region after the fertilization? Okay. So this is the zygote. It will be surrounded. It will be surrounded by a membrane called zona pellucida. So surrounding membrane will be called as zona pellucida. Okay. So the inner cell, it's a zygote. Surrounding the zygote, which I have represented in form of white coloration here, that is nothing but the zona pellucida. How about corona radiata cells? Yes, along with it, even corona radiata cells also will be present. But as the zygote starts dividing here, the corona radiata cells also will disappear. But for implantation purpose, only two components are important. One is the zygote, another is the zona pellucida. Okay. So again, you should know that this fertilization is going to occur in ampullary region. in the ampullary region of fallopian tube it is going to occur. So the zygote, the cell, unicellular cell zygote, which is represented here, so it is going to have, of course, the nucleus here inside. It is going to have the nucleus, zygote, and the zona pellucidum. Then this zygote starts dividing. So this is a diploid cell. Zygote is a diploid cell. The sex determination occurs at the level of zygote, and it will now start dividing. How does it divide? Is by a special kind of mitotic division occurs in the zygote. So special kind of mitotic division, kind of mitotic division, take division occurs in zygote. A special kind of, see, this zygote will divide. So special kind of mitotic division occurs in the zygote. So it has a term. That special kind of mitotic division occurring in zygote, it has a term, and that term is called cleavage. So what is this definition actually stands for? This definition stands for cleavage, a special kind of mitotic division occurring in the zygote. Okay. Next is within around 30 hours, division will complete. So this zygote which is there, it is going to divide. Okay. So divide and produce two cells. Divide and produce two cells. So now two daughter cells are formed. This cell is called as two-celled stage, it is called. So you can tell it is which cell stage? Two-celled stage. Okay. Then what are these daughter cells called? The daughter cells are called blastomeres. So zygote divides by a special kind of mitotic division, usually occurring within the first cleavage division occurs within 30 hours, and the daughter cells are called as blastomeres. The daughter cells are called as what? Blastomeres they are called. Now, as a result of cleavage of zygote, okay, first cleavage of zygote, two daughter cells are formed. They are called blastomeres. Now, how is cleavage differing from mitosis? Cleavage process is rather a rapid process. It is not a very slow division. Comparatively, it is a rapid process. Cleavage can occur rapidly. Then, since cleavage is a rapid, there is a lot of demand for oxygen, so there should be an increased requirement of oxygen. Increased requirement of oxygen. That means more oxygen is required. And when more oxygen is required, cellular respiration should increase. So also there is increase in cellular respiration. So also there should be increase in cellular respiration. Then what happens? Size of blastomeres are smaller. That is the daughter cells which are formed here. These blastomeres are daughter cells of the zygote. The size of the blastomeres are smaller than the parent cell. Next point, the size of the blastomeres are smaller than parent cell. Here zygote is a parent cell. It is going to divide and produce the blastomere, and always the size of the blastomere is going to be smaller than the parent cell. That is the nature of cleavage division. That's nature. Then next, what happens to the cytoplasm here? Cytoplasm becomes lesser and lesser after each cleavage division. And also, but the nuclear content is going to increase because the cells are doubling. The nucleus will never change. The content, the chromosome number and all will never, nature, the number, identity of all those chromosomes will never change. So as a result, what happens? The daughter cells will have, the daughter cells will have more nucleus, whereas less cytoplasm. Okay, compared to the previous structure here. So what was the nuclear content here and what is the nuclear content here? In both of them, it will increase. So size remains the same here. Two cell stage size is similar to zygote size. Okay. So therefore comes the word nuclear to cytoplasmic ratio will increase to cytoplasmic ratio is going to increase. Okay, this is also the nature of what? Cleavage divisions. Okay, nature of cleavage divisions. So two cell stages formed. Then this layer, this white layer, so I have drip like this. So this is the zygote, and surrounding the zygote will be what? Zona pellucida. So this is zona pellucida, and this will be the actual zygote. Okay. Zona pellucida still remains with the surrounding the zygote. Next, what happens is two cell stage becomes four cell stage, but it will become step by step. You can see that as division is occurring, the size is almost the same size like zygote because blastomeres are, there is no growth in blastomere, you can tell. No growth in blastomere because they are instead reducing in the size compared to the parent cell. So no growth in the blastomeres. Their size is not increasing. Not increasing. Their size is not increasing here. Okay. So no growth in the blastomeres. Now when this cell divides, what kind? How is the division? The division here is a holoblastic cleavage. The type of division that is occurring in human is holoblastic. First also holoblastic. Even in case of the second division, it is holoblastic cleavage only. So what is holoblastic cleavage? One cell will divide completely. Okay, divide completely. When division is complete, complete division is occurring, that kind of division is called holoblastic cleavage. Okay, that means here again definition, complete cleavage or complete division. Cleavage is a special kind of division here. So complete cleavage. Complete cleavage occurs. So, in some animals like insects or birds, only superficial division is there. The zygote will not divide completely, but it will divide only partially like this. Whereas rest part will be not dividing. Okay, that kind of division is called the meroblastic here. So otherwise, holoblastic, which occurs in human, it is nothing but a complete division. When complete division will occur means two cells are now separated. They are two different means different cells now. They are not uh, a single cell with which is partially divided, but two separate cells are formed due to holoblastic cleavage, which is nothing but complete cleavage. Now, how does the pattern of cleavage depend? Sometimes in some animals complete division is there. In some animals uh, there is no complete division there. Holoblastic cleavage is not there. So pattern of cleavage ultimately depends on your content. So pattern of cleavage depends on yolk content. Human being egg is L-acetal, you can tell the or micro L-acetal because the yolk is very less in human egg. When yolk is less, that means if a egg have very small amount of yolk, then it will not create any problem in the division. That means complete division will occur or holoblastic division is going to occur because of less yolk content. It is not going to pose a problem for cell division. Whereas certain eggs are very rich in, say, birds like that. So they are rich in yolk. So yolk content is more. Okay. When yolk content is more in a cell, in a fertilized egg, when yolk content is more, this cell now can't divide completely because it becomes too bulky for the cell division to occur when yolk content is more. So what happens? Instead, the cell will divide only partially like this. So partial division of the cells will occur. Okay, but not the complete. Okay, but in human, because of microlecythal or alecythal condition, the division is you can also alecythal condition. The division is holoblastic. Therefore, conclusion will be pattern of cleavage depends upon the yolk content. Okay, then two cell stage becomes four cell stage. Now in human beings, how does the two cell stage becomes four cell stage? Is asynchronously rather than synchronously. For example, here there are already two cell stage formed here, two cell stage. So two cell stage at once will not become four cell stage. So instead what happens, a division occurs and an intermediate three cell stage is formed, and then finally division occurs, and what occurs, four cell stages formed. Okay, step by step it occurs. So if I say two cell stage will at once form four cell stage, then it is called synchronous division. Then it is called as the synchronous division. But instead, two cell stage forms an intermediate three cell stage and then it forms the four cell stage. Then it is called asynchronous. Asynchronous division. So in human, basically 3-cell intermediate is formed. So asynchronous cell division is present. So therefore 2-cell stage forms 4-cell stage through intermediate 3-cell stage. Okay. So this was first cleavage and this is going to be the second cleavage. Now 4-cell stage will form 8-cell staged. Four-cell stage forms 8-cell stage. Okay. Then 8-cell stage further division occurs. One parent cell divides into two. So 8 parent cell means it will divide into two and form 16-cell stage. So 8-cell stage further forms 16-cell stage. For the forms which cell? 16-cell stage. Now 8 to 16-cell stage will look like a fruit, mulberry fruit, when observed. When it is observed microscopically, it is going to look like a mulberry fruit. Okay. And since it appears like mulberry fruit, it is having a name there. And that is why it's called morula. What is morula? The structure that appears like a fruit called mulberry. Therefore, 8 to 16 cell early embryo is called morula stage. It should be between 8 to 16 cell stages. Can be even more also, but typically between these two range, it can be called as a morula. Okay. So around in 4th day, the morula is formed and this structure will move in the fallopian tube. How does it move? The fallopian tube on inside is going to have columnar epithelium and which is going to have the cilia arising from the basal body. So this ciliated columnar epithelium will be lining the fallopian tube. So beating action of this cilia, which is also called ciliary movement, because of which this structure helps this structure to move. And that movement can also be sustained or further supported by peristaltic movement of the fallopian tube also. So morula stage. So after morula, the morula will now enter into the womb. Now this part, this is called the womb or uterus. The other part is quite symmetric. So only the one part I have drawn. This is nothing but the womb or the uterus. It is the womb or the uterus. So the morula enters here in the uterus. It is again almost the same size. Now you should know it is not increasing in size. Here it is as such not increasing in size. It is almost the same size of that of the zygote, remember. Why? Because of the nature of cleavage. What is the nature of cleavage? Daughter cells are smaller than the parent cell. So even though a single zygote has formed 16 cell stage, those 16 daughter cells are much smaller than their parent cell. As a result, the size of morula is same as the size of zygote due to this nature of cleavage. Okay, embryo like morula here is the same as zygote. Okay, the size of early embryo, that is the morula, is the same as that of the zygote. Why? Because of nature of cleavage. Nature of cleavage. Understand? So zygote size, morula size is the same. Okay. The morula will now roll into uterus, 16 cell stage. Those all small daughter cells, 16 cell stage. They move, or you can tell roll in the uterus. So they're not directly dropping, you can tell. You can tell they're rolling in the uterine wall, over the uterine wall. The morula will further undergo compaction. So as it moves to the blastocyst, as it forms here and then moves to the blastocyst, it can undergo a phenomena called compaction, where the intercellular space will decrease and the cell will become more compact and compact. Okay, and that phenomena is called as compaction. That phenomena is called as compaction. During this compaction, usually the flat cells will be on the surroundings, like they start rearranging themselves, which I'll explain further how the morula will start forming blastula. Okay, then this morula, the cells in this morula further starts increasing in number and then form next embryonic stage, and that is called blastocyst. How does that occur? So this is the morula, which is having dividing cells inside, 8 to 16 or more also, basically 8 to 16. So further divides from 16 to further doubles, it doubles up. Now this one, this structure was called as what? Morula. The morula will undergo cell rearrangement occur. That means cell will start arranging themselves in different order. So the morula cell rearrangement occurs. When cell rearrangement occurs, what happens? The flat cell starts coming at the periphery like this. So flat cells will be at the periphery. Of course, they are all nucleated cells. And some of the cells remain attached to these flat cells. That means these cells inside, they will arrange themselves and form a structure, the next early embryonic structure called blastocyst. Next early embryonic structure called blastocyst. This phenomena is called blastulation. This whole process or the phenomena where morula is converted into blastula. Now I will tell what is the difference between a blastula and blastocyst. Mammalian, other animals, the term called blastula can be used, but here in case of human, you can use the word human blastula is called blastocyst. Since human blastula is being referred here, it can be called as blastocyst. Now this process is called blastulation. So that transformation of morula and blastocyst occur here. Okay. So what is formed? Blastocyst is formed, or this phenomenon called blastulation occurs. Now, as this rolls into the uterus, the secretion of this uterine secretion, that is usually secreted by the uterine glands, the uterine secretions of this uterus is going to dissolve, is known to dissolve the zona pellucida. So this now the surrounding was zona pellucida, and inside, what is this? Flat layer called, and the inner cells. Okay. Now the zona pellucida starts dissolving. When zona pellucida starts dissolving, then blastocyst starts coming out. Blastocyst starts hatching out, you can tell, or coming out, because the zona pellucida is dissolved, it is disappearing in the uterus due to secretions of the uterus. So this is called zona hatching. This phenomenon is called as zona hatching. So therefore zona hatching occurs in the uterus. Got? So now what occurs is the blastocyst comes out of zona pellucida. Okay, that phenomenon is called what? Zona hatching. Okay. Next is structure of blastomere. I'm going to explain the structure of blastomere. So this is the typical structure of blastomere. Outside it is going to have flat cells. Outside is going to have flat cells. These are called trophoblast cells. Whereas certain cells remain poke into this cavity called blastocell, or they protrude in this cavity called blastocell. How did all this form? Due to cell rearrangement from morula. So all the cells that were present in morula, they started rearranging and then forming the cells of blastula. Structure of blastula, three main structures will be here. The outer thin layer, this is called trophoblast layer. This is called which layer? Trophoblast layer. Then there is a cavity here. It is called blastocele. It's called as blastocele cavity. This cavity is formed in blastocyst. And this is inner cell mass. Inner cell mass. Now first, trophoblast. Trophoblast is the outer layer of thin cells in blastocyst. In future, this trophoblast in future, it develops into extra embryonic membranes. The known extra embryonic membranes are what? Chorion, amnion, allantois, and yolk sac. These are the 4 extra embryonic membranes. chorion, amnion, allantois, and yolk sac. These are the four extra embryonic membrane, which will be basically further developing with the help of this. I mean, developed from the trophoblast. Okay. So out of this function, chorion, very important for formation of placenta further. Amnion, it is a cavity where fluid called amniotic fluid is present. Allantois, this is going to, in human, it does not have much role, but it is going to develop into certain blood vessels in the umbilical cord. And yolk sac, in early stages, it will be, which is present in early stages, it will be important for hematopoiesis or hemopoiesis, you can tell, that is production of blood cells. So it's a site for production of blood cells. So trophoblast is responsible for extra embryonic membranes. Why? Because as embryo develops, to protect and support that embryo, surrounding that embryo, these extra embryonic membranes should be there, and that is going to be developed from the trophoblast. Okay, however, this cavity is going to obliterate. Okay, then next comes the ICM. ICM is inner cell mass. It is also called embryoblast. So embryoblast it is also called. It's also called embryoblast cells. As the term indicates, it is responsible for formation of embryo. That means different parts of the body. These cells will undergo cell differentiation, cell division, okay, and therefore gives rise, and then gives rise to different organs and tissues of the body. So whatever the body parts are there, different body parts are there, they are basically developed from the ICM. So broadly, what you came to know here is this will be producing extra embryonic membrane. Those membrane further can develop into placenta and all, like chorion. And this can form what? Extra embryonic membrane. This will form the actual embryo, that is the body parts. Various body parts, the organ systems, and all the body parts will be formed from this ICM. They have a term. Trophoblast can also be called trophoectoderm. Trophoectoderm. That will be like when further the next ICM, when further steps will be taught. So here next will be ICM, that is also called embryoblast. Okay, ICM cells are more or less like stem cells. More or less like stem cells. So that is how, what will be there? Different parts of blastocyst is going to be there. But why it is called why it is not called blastocyst? Why cyst? It is because this ICM will poke inside this cavity as a cyst. Okay, as a cyst, and that's why it's called as a blastocyst. Okay. So this is how what is formed as blastocyst is formed. Now the blastocyst will hatch out of the zona pellucida. It means that the zona pellucida will dissolve due to secretions of uterus. Now blastocyst, that is the outer layer trophoblast, will start differentiating and forming two layers. The outer syncytiotrophoblast and the inner cytotrophoblast. Two layers are, differentiation of two layers occur. Now, as the blastocyst will come in contact with this endometrium, changes occur in this endometrium. Okay, the epidermis starts disintegrating. Then fibroblast cells present there will start differentiating and forming the large size cells which are rich in glycogen, and it becomes nutrient rich. This part becomes nutrient rich, and this part also becomes highly vascularized. That's how changes will occur. Now, what happens here is the trophoblast will start secreting lytic enzymes. Okay, they start secreting what? Lytic enzymes, which are basically hydrolytic enzymes only, that will break have an ability to break. Those lytic enzymes will start dissolving this endometrial part. Okay, and what happens is a pit is formed here. A pit is going to be formed. It is an edematous pit. So inside that pit, what is going to get buried is blastocyst is going to get buried. Okay. And then what happens? The cells of the uterus will start covering this buried blastocyst. Okay. And thus what completes is implantation completes. Okay. And during that time itself, after implantation, now this is not called as an endometrium, it is called as decidua. What occurs is decidual reaction occurs once implantation will complete. Now decidua is nothing but the the thickening of the endometrium after implantation. The endometrium after implantation is called as decidua. Okay. So this one at the base, it is called decidua basalis. The one, what is decidua basalis is the trophoblast, which is differentiated into two layers. Okay, syncytial trophoblast, they start secreting lytic enzyme, also produce finger-like projections, which in future forms chorionic villi, and later on will be continuing being part of placenta. So this is later on continues to be part of placenta. Okay, chorionic villi will continue and form placenta. So here, this is the finger-like projection formed from the trophoblast. Okay, the wall which is present here, this part of the decidua is called as decidua basalis. This is called as decidua basalis. And the part of endometrium that has covered this blastocyst on top, not at the base here, on top, that is called as decidua capsularis. So it means that in the pit, the blastocyst is buried. When uterine wall covers that, the upper part, that part that covers on top, that is called as decidua capsularis. Decidua capsularis. It is called as decidua capsularis. Then the endometrium, which will not be at the base, or neither it covers the rest of the endometrium, that is here, here, and the rest part here, that is called decidua paritalis. That's called as decidua paritalis. That is called decidua paritalis. That means the endometrium after implantation is further differentiated into decidua paritalis, capsularis, and basalis, depending upon whether it is very much near to the blastocyst or away from the blastocyst. So this completes. For this implantation, it will be occurring in 7 days after fertilization, the time required for implantation. It will occur 7 days after fertilization. So in short to tell, when zygote is fertilized in ampullary region, it will undergo special kind of division called cleavage to ultimately form morula. Morula remains in fallopian tube and slowly it will come in the uterus. So in the uterus, it will undergo blastulation and a structure called blastocyst is formed that is going to bury in the endometrium, thus transforming this endometrium into decidua. Okay, when you call it as decidua is when decidual reaction occurs. Okay, then the region becomes more thicker, more vascularized, more nutrient rich. Those that will be the decidua. Okay, so what completes here is implantation completes, and this process is going to take 7 days after fertilization. So here how thick it becomes, so it may reach to 14 mm as against 5 mm in luteal phase or as against just 3 mm in the follicular phase. So thickness will keep on increasing. So that itself is about the implantation. So what happens is after implantation, for example, this is the say blastocyst with ICM. The blastocyst will form 3 germ layers: ectoderm, mesoderm, and endoderm. So this ICM cells will rearrange, they undergo migration. This is called as morphogenetic movements occur. Morphogenetic movements. Morphogenetic movements occur. Due to this morphogenetic movements, what will happen? 3 germ layers are formed, and that structure which contains the 3 germ layers is called gastrula, and the phenomena is called as gastrulation. Before formation of gastrula, there is an intermediate 2 layer called 2 layers will be present, and that is called bilaminar disc, consisting of epiblast and hypoblast, which will later on form these 3 layers due to morphogenetic movements. Some of the examples of morphogenetic movement is in invagination, ingression, okay, then emboli, delamination. These are some of the examples of morphogenetic movements. But these movements are not just in humans; they are present in gastrulation of broadly different animals. So therefore, this is example of how what occurs. This example of early embryo, and even blastocyst is an example of early embryo. Okay, so this completes implantation. Further topics will be continued.